Good morning. Today we are discussing the biostatistics. What are the main objectives? What are the main objectives of the biostatistics? Uh, Understanding concept of the probability and its rules in the applicable of the genetics. Use the chi-square test in the goodness of the fitter relationship genetic ratios to calculate the arithmetic mean, variancy, standard deviation, standard error for the quantitative characters to compare the two samples by the testing for equality of their the means through the t-test. The participation viability, the next one is the through the analysis variancy. What are the introduction of the statistics? Statistics means the measured, the cofactor, and piece of information stated with the figure such the height of the one person, birth rate of the statistic. Statistics, they also called as the data, is the purely started the more than one figure such as the height of the two person, birth of the two five babies. They are the statistics. Mainly, who is the father of biostatics? Is the Francis Galton is the father of biostatistics. Definition of the biostatics. Biostatics means when we are mainly the statics also they can be used for the many fields like the medicine, medical statistics, agricultural statistics, economical statistics, mathematical statistics, and so and so. Definition of the biostatics. It means when we use the statistics tools on the biological problems and derive some results about the example medicinal science. It is also called as the biometry. It means measurement of life. Normally in medicine, for the precious, appreciation and the facts, observation or measurements have to be the expressed in the figure biostats. What are the uses of the biostatistics? It calculate an average, median, mode. Is a branch of the statistics that applies the statistical method in the biological, medical, and health related research. It helps the designing experiment, collecting data, analyzing results, and the drawing meaningful conclusions in the fields of medicine, public health, and genetics and the epidemiology. Key areas of the biostatistics, descriptive statistics. Descriptive statistics summarize and describe the main features of the data is in the common descriptive measurements include. Okay. What are the main uses of the biostatistics? To calculate average, median, mode, and standard deviation of the given collected data. To compare the two sets of the data. To get conclusion or results. To find the association between the two variables. To find the correlation between the two variables. To give the results in a tabular or the diagrammatic form. Biostatistics. But is in public health and community health is also called as the health statistics. The medicinal also they can relate to the medicinal statistics. Okay. What are the measure? Measurements of the scale, the variable. A variable is a characteristic that the varies from one biological entity to another term is the variable. Various ratio scale. The next one is the internal scale, ordinary scale, normal scales. These are also they can use for the cell. Types of the data also there are the two types: qualitative data and quantitative data. Qualitative data, especially nominal and ordinal, and next one is the quantitative, descriptive, and continuous. These are the data. Mainly the statistical method applied with the biological problem is also called as the biostatistics. It is also called as the biometry. Biometry means is the biological measurements of the or measurements of the life, or especially as life sciences. The statistical method, the Croxton and the Gordon said the statistical method means the collection, presentation, analysis of the interpretation of the numerical data, collection of data. In this, the first step in the collection of data, careful planning and essential before the collecting the data. This one presentation of the data. These are the aspects. The mass data collected should be presented in the suitable from the further analysis. The collected data may be presented from the tabular or diagrammatic and graphical form. Analysis of data. The data presented should be carefully analyzed from the presented data such as the measures of central tendency, discrepancy, correlation, and regulation. Statistical methods mainly interpretation of the data is the statistical method. Mainly the final steps drawing conclusion from the data collection, avoid the conclusion must be drawn on the basis of the analysis. In higher degree skills of and the experience is the necessary for the interpretation. Measurements of the central tendency. These are the divided the three types. One is the mean. Mean is also called as the average. Median, the positional average, middle value. Most repeated value is the frequent value. Is the central tendency. Okay, central tendency measurements of the central tendency, mean, median, mode. And measurements of the dispersion. Next one, measurements of the dispersion. dispersion, and next one range, variance, standard deviation, and data visualization graphics. And next one, the next one, central tendency. Yeah, I will discuss the each and everything. Mean also is the average is also called as AM. Mean can never is the bias. It is also what are the merits of the mean and demerits of the means. It can be never be biased. It is the easy to calculate. It is the least affected by the influctuations for the sampling. It is based on all the observation of the series. Therefore, the most representative in variancy measure. What are the demerits of the mean? It is greatly affected by the extreme fluctuations. Thus, it is not a true representation value of all the items in the series. Aim cannot be used the qualitative characters such as the color, flowers, and stress and orange darknesses of the color. Especially these are the quantitative characters. They can measure by the mean only quantitative characters. The next one is the median, middle value. What are the merits of the median? Median is also easily calculate and understood. It is not affected the extreme observation. It is the best measure of the qualitative data. Only in mainly mean also they can measurements of the only quantitative data. Okay, these are also mainly they can best measures of the quality data median by the median. What are the demerits of the median? It cannot be determined in the case of the even number of observation. We merely estimate as the arithmetic mean of the two middle terms. This is the positional average. It cannot be accepted to the each and every observation. Mode what are the mode merits of the mode? It is easy to calculate and understood. It is not affected the extreme observation. It can be calculated from the grouped frequency distribution. Main mode mean the repetitive value. Mode is the repetitive value. Okay, mean mode. Next one is the range, standard deviation, and next one is the measurements of the variability or the measurement dispersion is the range, standard deviation, mean deviation, quartile deviation, variation, coefficient variation. Okay, correlation, types of the correlation, positive, negative correlations. Mm. Probability theory. What is the meaning of the probability? Probability is the plays the vital role in the biostatics, especially predicting of the li- likelihood and the livelihood and the health outcomes. Probability disturbances normally and binomial position disturbancy. And next one is the probability is based on the new evidence. What is especially probability? What are the basics of the probability? Probability measures likelihood and event in occurring and ranges from the 0 to 1. Key of the probability sample space, the set of the all possible outcomes and the experiment. Event E. Probability equation is the PE equal to number of favorable outcomes by total number of outcomes. Next one is the probability event is the P equal to number of favorable outcomes by total number of outcomes. Types of the probability, classical probability is used when the all outcomes are the equally likely. Next one is the empirical probability based on the observed data. Okay, the problem empirical probability, and subjective probability based on the experiment opinion or belief rather than the data probability. Next one, probability distribution in biostatistics, descriptive and the probability disturbances is it when the variables take specific countable. Okay, next one is the probability distribution in biostatics. Descriptive probability disturbances is the variable takes the specific countable values. Binomial distribution used the okay, binomial distribution used the binary yes or no. Outcomes is the infected or versus not infected. Formula F is equal to n PQQ 1 minus P by n minus 1 and number of traits. Next one is the position distribution and the used the rare events occurring the over time or space. Formula P is equal to x by key is x by the key. The example the predicting the number of new COVID cases per the day in the hospital. Continuous probability used for the variables that can take any value within the range. Blood pressure levels in the survival times. Normal disturbance is also called as the Gaussian disturbance. A bell-shaped curve common in the biological data defined by the mean uh, mm, delta n standard deviation. Okay, the exponential disturbance used for the time to event analysis. That is the example time until the patient recovers from the illnesses. Probability theorem in the biostatics. Bias theorem, conditional probability used in the diagnosis and disease screening to update the probabilities based on the new formation probability. Application of the probability in biostatistics. Next one, some of the probability rules in the product law are multiple. multiplicative calculate of the rule, and this is sum law and additive rules, conditional probability, the application of the binomial theorem in the probability. What are the application of the probability? Mainly is that the disease risk prediction, estimating and probability, estimating the probability of the developing of a disease based on the risk factors, diagnostic test accuracy, evaluating the test sensitivity in the specificity, positive predictive value and negative predictive values. Next important is the survival analysis, estimating the patient survival rates in the clinical trials, in the epidemiology, predicting the spread of infectious diseases using the probabilistic method models, clinical trials, determining the probability of the treatment success in the randomized the controlled trials. What are the conclusion of the probability? Probability is essential in the biostatics for the making data-driven decisions in medicine and public health. It allows the research to access the risk, predict outcomes, and the design effective studies. Next is the chi-square test. Chi-square test of the goodness of fit. Chi-square test is the chi-square test, the goodness of test is the statistical test used to determine the whether a sample distribution matches an expected distribution. It is commonly applied in the biostatics to analyze the categorical data such as a disease prevalence, genetic disturbances, and the clinical trials outcomes. When to use the chi-square goodness of the fit test, that this test is the appropriate when we have the one categorized variable with the two or more levels. You want to compare the observed frequencies with the expected frequencies. The data consists of the counts or the frequencies, not percentage or continuous values. Hypothesis in the test, null hypothesis, the observed frequencies follow the expected disturbance. Null hypothesis, the observed frequencies follow the expected disturbance. Alternative hypothesis, the observed frequency do not follow the expected disturbances. For example, we expect the equal proportion of four blood types in the population, but the observed data differs. We test whether the deviation is due to the change or the significant differences. Next, what are the formula of the chi-square test? X square equal to sum of O minus E square by E. O mean observed frequency. O mean observed frequency. E mean expected frequency. Two-way ANOVA, definition of the two-way ANOVA, test effects of the two independent variables. One dependent variable, it is also checks the for interaction affects the between the factors. Two-way ANOVA test, the main effect of the diet, main effect of exercise, yes or no. Interaction effect, does exercise influence diet factors? Repeated measures of the ANOVA. Repeated measures of ANOVA compares the mean when the same objectives are the tested under different conditions or multiple times of the point. Post hoc test for the significant ANOVA results per case